everyone. I'm sorry that I won't be able to join you guys on Monday afternoon. It's currently Saturday and I'm recording this video ahead of time. On Monday afternoon, I'm going to be out with my husband for some medical tests. So I just want to be prepared for lab. So again, I'm going to make this video ahead of time so that you'll be able to do lab like normal. If you have any questions, feel free to shoot me an email after lab, stop by on Tuesday or later on in the week, and I'll be happy to talk you through anything that might have come up during the lab session. All right, so for now, I want to get on to the topic for the week. So there should be a pile of handouts up front on the table. Ask the TAs to put them there. And I know it's not the same over video, but I'll do my best. I have to figure out where to hold this. Okay, so we're studying flatworms this week. There are, as you can see on the handout, there are 3 types of flatworms. You've probably heard of the tapeworms and the flukes. You probably haven't heard of the planaria. The reason that you've probably heard of tapeworms and flukes is that they are parasitic. They cause disease. Tapeworms cause all kinds of digestive issues. They're often associated with undercooked meat. They can grow really long, like 12 feet long. So you wouldn't want something like that growing in your intestines. Flukes cause a disease called schistosomiasis. It's one that we don't hear of a lot in the United States. It's more common in tropical areas. And it is worth noting, though, because it is the second most common parasitic disease, second only to malaria. So worldwide, it's a major cause of illness. Planaria are the 3rd type of flatworm. That's what we're going to be working with today. They don't cause disease. Although, still, be careful with them. Don't eat them or do anything stupid. And anyway, so we're working with the planaria, and they'll be our model organism. You're going to design your own lab this week to test some kind of a behavior of planaria. Okay, so at the bottom is just some basic background on planaria. They're really small. Again, there's no scale bar on the picture there, but small, I'd say like 1 to 2 centimeters. So very small little worms. You're welcome to look at them under the dissecting scope to kind of see their details up close. They do have bilateral symmetry. They don't have a respiratory system or circulatory system, so they're pretty simple creatures. One of the things I want to note on the picture here on the left-hand side is that there's only one opening. So there is a body cavity, and so things can come in and go out of the worm. But that one opening serves as where the food's going to go in and where the waste is going to come out. So it's kind of a unique setup. Now, I just mentioned that they don't have a respiratory system, but they need oxygen. And they get oxygen simply through diffusion, which is why they have to be flat, and hence the name flatworms. So again, they're going to respire and get their oxygen simply through diffusion. All right, so on the back, we're going to go into a little bit more detail about why people study planaria. There's lots of worms out there. Why planaria? And there's a little box here that says they're a great model for behavioral biology. So one of the key points there is that they're a very simple animal. They are invertebrates. And they're one of the simplest animals that have a bilateral cephalized nervous system. Okay, so let's put that in everyday language. That means in the head area, they have cerebral ganglia. You can see that on the picture there. It's basically a primitive brain. So they're able to process information. They have nerves, which are bundles of axons, and you can see those flowing through their body, basically long way through the body and across their body. They also use mammalian-like neurotransmitters, which means some things that we can learn about planaria have applications to other organisms as well, which is important because most people don't care just about planaria. Probably the last point here is one of the most important reasons that they're used as a model organism. They're one of the simplest animals that can learn in response to conditioning. So they're often used for behavioral biology studies. Okay. So again, today, you're going to design your own project around some kind of theme to study behavioral biology. So I know from the syllabus of 182 with Professor Devoto, you've been talking about homeostasis, thermoregulation, osmoregulation, I believe. And that's why I'm pausing because I'm not actually sitting in on those classes, but I know you had those things listed on the syllabus. And so you're welcome to delve into one of those areas if you want to explore something that you've covered in 182 and see how that plays out with the planaria. Or You're welcome to pick any topic of interest to you. I'm going to go through a couple examples, but again, if you have a cool idea, go for it. Have fun with this lab. Design your own labs are a lot of fun because you get to do whatever you want. Sometimes things work, sometimes they don't, but be creative. That's another nice thing about this class being pass-fail. You don't have to worry about getting beautiful results. Let that scientific creativity out there. Have some fun. All right, so down at the bottom, there's a picture of the planaria, and there's an area there where there are chemosensors. So some of you, I guess I should hold this where you can see, there's an area there where there's chemosensors. Some of you might decide to test different chemicals and see if the worms, if or how the worms respond to those chemicals. Maybe they like certain things, they move towards them, maybe certain things make them move faster. Those are the types of questions you're going to be asking. So in the lab area, so you're going to go to your lab room, and then in that back prep area, there's going to be a cart with all kinds of chemicals. You're welcome to use any of those to make up solutions. You could make something with sugars, we have salts, acids and bases, I put some caffeine out. There's all kinds of just the basic lab chemicals we have. There's also a little bit of the neurotransmitters that were left from last week. So again, you're welcome to make up chemicals. In thinking about chemical solutions, you would want to make them in what's called Planaria water. So on your lab bench today is a big jar of Planaria water. If you're making solutions, make sure to use Planaria water. Now, I labeled it Planaria water. You don't actually buy Planaria water. What it truly is aquarium water. We need water that's pH balanced, has the right osmolarity. We don't want to use tap water because tap water has chlorine and it'll kill the worms. So again, anytime you're making solutions or working with the worms today, make sure you're using Planaria water. Okay, so you could use, or you could do an experiment with chemicals. You could test different concentrations, different types of chemicals, again, whatever. You'll see in this picture of the planaria, there's also an area where they have photoreceptors. So you might design an experiment looking at light. How do they respond to light? So if we think about light, off to the right, I have a picture of the planaria's eye. So there's a little box there that's a blow up of the eye. You'll see it has photoreceptors, there's some pigment in there. One of the big things that's missing is the lens. So a key point is that if there's no lens, the planaria can't actually focus the light. They're going to be able to sense light, so you can see maybe if they can sense intensity or directionality or different wavelengths or colors or experiments like that, but just know that they can't actually see you or see images like we typically think of when we think of the sense of vision. I'm trying to think of some other common experiments you could do. Temperature could be a really cool one. I guess that's already listed here under the idea of thermoregulation, but you're welcome to use anything in the lab. So for example, each lab has microwaves and fridges and freezers. I did put some planaria water in the fridge if you wanted to use water that's already cold, see different temperatures, if or how drastically does that affect the planaria's movement. And if you have another creative idea and you want to go get supplies from your room or from the cafeteria, please feel free to do so. Okay, so you'll design your project. Jump to the next page. Before I get to experimental design, I just want to mention what I have on the top of the page there. We're using planaria. You might think, okay, these are stupid little simple things. Like, is this, do people in real labs use planaria, or is it just something we're doing because it's intrabiot? And what I am trying to stress is that people in real research labs use planaria too. So I pulled out three actually not the abstracts, just the titles of recent research articles that are using planaria. The one on the top is from November of 2019. So again, these aren't from the 60s or 70s. These aren't ancient. People currently are using planaria. And the top article is talking about how calcium ions in different aquatic environments are driving planarias to food. Again, there's two other articles there. I put all three of those on Moodle if you want to check any of them out. Planaria are also used for a lot of environmental studies to look at toxicity of chemicals. They're also used for regeneration purposes, although it takes longer than a couple hours to regenerate, so we can't study regeneration in our lab section. All right, so people use them in research labs. We're going to use them in our lab today. So experimental design. And you can deviate from this a little bit, but I want to give you kind of a basis to start with, so you're not starting completely from scratch with no clue what to do. All right, so in your lab is going to be a whole big pile of these Planaria grid sheets. Okay, and in the picture there, I have 2 of them, and I did that for a purpose. You're going to have 2 of them, and you're going to lay them flat on your bench. Okay. One of them, you're going to have a big petri dish on it, and you're going to put solution in it. And then that circle right in the middle, you're going to drop your worm there, and then you're going to watch the worm move around the dish. We'll get to some more details. And then the second grid sheet is so that you can make observations and like use a Sharpie and like draw the path or write times down. Clearly, you can't do that all on just one, so that's why you're going to have 2. One where the actual experiment is happening, and the second where you're recording your data. Now, there's 2 general ways to set up the experiment. One is to use, it's awkward here, to figure out how to put stuff. Okay, so one is to use a fixed stimulus. The other is to use a non-fixed stimulus. What a fixed stimulus is, it simply means that whatever you're testing is in a certain spot on that grid sheet. So typically, you would put that kind of at the top of the grid sheet. So your worm you'll put in the middle. Maybe you'll put a sponge that's saturated in a certain chemical at this very top. And maybe the question you're going to ask is, does the worm swim towards that sponge that's saturated in your chemical? Or does it swim away from it? Or could the worm care less? Okay, so you're basically looking at, in that example, chemotaxis. Again, are the worms moving towards it, away from it, or are they neutral towards it? So if you're thinking of a fixed stimulus, you could use that example of sponge saturated in chemical. You could use current. You could use a light source and shine some kind of a light in a certain spot and say, do the worms move towards it or away from it, or not at all? Or I guess they'll probably move at all. If they don't move at all, it might be dead, so look at any worm. But if they just seem to be moving randomly, it could be that whatever your stimulus is doesn't have a true effect. Okay, so you could have a fixed stimulus. Another way to set things up is to use a non-fixed stimulus. So certain things would affect the whole dish of Planaria water. So for example, if you're doing temperature, you can't really just make the water at the top end of your dish warmer. It just wouldn't work. All of the water would be a certain temperature. And so what you would be seeing is you'd have your dish, you'd have it filled with water, and you'd be asking the question, at a higher temperature, do the worms move more or faster compared to when the water's at room temperature? Okay. Again, you'd have your one grid sheet where you're doing an experiment, another one where you're making your observation. But with a non-fixed stimulus, those positive, neutral, negative areas, they don't matter. They're there in case you were doing the other scenario. But they don't matter for a non-fixed stimulus. Again, you could do temperature, which would affect every bit of water in the dish. You could also make a chemical and just dump it in the whole dish. So you could make a certain sugar solution, put it in the dish, and say, okay, maybe your hypothesis is sugar is going to make the worms happy, and so they're going to move around a lot more compared to the control, which is just regular water. Okay. So pre-treatment with the chemical solutions throughout. Oh, pre-treatment would mean perhaps you're going to soak the worm in the solution first and then put it in the dish to test it. So there's all kinds of variations and tweaks on these things I'm mentioning that you could decide to go with for your particular project. All right, now overall, each trial, so each time you're working with a worm, I would recommend doing 5-minute trials. There are situations where you might want to do shorter or less time, but as a default, go with 5 minutes. You're just going to do one overall project today. It should have a control, and it should have 2 experimental variables, and there should be a theme to your project. So don't do one temperature and one salt like that. There's just no central theme there. So if you're going to do a temperature experiment, maybe do room temperature and then 2 other temperatures. So have a theme to all kind of 3 scenarios you're working with. Again, if I had a chalkboard, I'd write this up there. So you're going to have control, condition 1, condition 2. And that's your entire project. Now, everyone's worm is going to be a little different. So you don't want to just do each of those one time. You want to have at least 3 replicas of each condition. So that means you're going to do the control 3 times. You're going to do condition 1 3 times. And you're going to do condition 2 3 times. So at a minimum, you're going to need 9 worms. And you should use a different worm each time. If you keep using the same one, it it won't work. They might get tired. They actually might get damaged as you're moving them around. So again, use a fresh worm for each trial at a minimum 9, but we have tons of worms. So you're not limited in terms of worms. There's going to be a whole jar of worms for you to use. So if you want to use 30 of them, that would be okay. All right, so again, control 2 experimentals and then 3 replicates of each. All right, so last page. What I'm going to talk about on the back is what kind of data you want to record in that data logging sheet. In the lab manual, I present a couple different possibilities. So actually on page 27, I list A, B, C, D, and E as 5 different things you can measure. I'm actually going to focus on 2 that I think are particularly easy and meaningful. So right now I'm going to focus on, if you're using a fixed stimuli, looking at a certain parameter, and if you're using a non-fixed stimuli, looking at a different type of parameter to measure. And again, you're only going to do one of these, but you can decide what makes the most sense based on your particular experiment. So if you have a fixed stimuli, something you might do is calculate the % of time that the worm is spending in the positive area, neutral area, and or the negative area. So overall, let's say you have your grid sheet, put your worm in the middle, you put a light source up here at the very top, and you're saying, okay, I shined a happy yellow light, yellow smiley faces, maybe you think the worms are going to like yellow light. And so you're going to do the experiment, you're going to put the worm here on your data sheet, you're going to record where the worm goes, and then you're going to calculate the % of time that the worm spent in that positive area. And maybe what you find is initially the worm went up there. So at time 5 seconds it went up there, and then at time 15 seconds it crossed this other line. So again, you're going to be drawing the path and noting those times when it crosses between positive and neutral. And then maybe it went right down to the bottom and then stayed in this area for the next over 4 minutes. So 5 minutes, 5 times 60, that would be 300 seconds altogether. So maybe it spent 10 seconds up here and then almost all the rest of the time was spent down here. So that would mean that the % of time spent in the positive area would be 10 seconds out of a total of 300 seconds. So you can do out the math and find the % of time that the worm spent in the positive area. Again, you would do that for all of your replicates, and then you would compare that data to the data for your control as well as for your other condition. That might have been a lot to take in just with me holding it up and talking. So I put a sample sheet with it drawn out and the calculations done on the chalkboard in your lab room. So if you end up doing this scenario, definitely check that sheet I have up on the chalkboard in your lab room. Again, it's written out. Hopefully it'll be a little clearer there. All right, now if you're doing a non-fixed stimuli, positive, neutral, and negative areas, that doesn't really mean anything. So if you're doing a non-fixed stimuli, I recommend that you do something called PLMV, which stands for Planaria locomotor velocity. It sounds pretty scientific. All it is the number of grid lines that were crossed. Okay, it's kind of funny because I wanted to show you that I didn't just make this one up. So this is a figure from a research article where they're measuring PMLV. Again, it sounds technical. All they did was if you look at that article, they had a grid sheet very similar to this, and they counted how many times did the worm cross one of the walls, one of the grids. And that's their measure of how active the worm is. So if the worm is just kind of sitting there still not doing anything, it's going to cross maybe 10 lines. If it's super active, maybe it's going to cross 40 lines. And so this is a way to quickly measure how active is the worm, how many grid lines is it crossing. So again, that's a very simple way to get good data if you're using a non-fixed stimuli. Again, if you don't like either of those, there's some more in the lab manual you can choose from. All right, so you're going to collect your data. Again, think about that ahead of time so you know what to record, how you're going to record it, make sure everyone's using the same units, etc. Another thing I should mention is you're going to be working in, there's going to be 4 groups in each room. It's probably the best way to say it. So within your group, you're welcome to divvy up the work. So I would recommend for the first 2 or 3 trials, everyone should work together so that you understand how to do it and everyone is doing things the same way. But then after that, divvy up. So then maybe someone else is going to repeat all the replicas of this one and someone else is going to repeat all the replicas of this other one. So you can basically have 2 or 3 people working at one time, and that'll help to speed up the process of actually getting all your replicas done. All right, so you're going to collect your data, and then next week there's going to be a poster session. I'll talk more about the poster at the very bottom, but on your poster you're going to be asked to put a stats table like the one I have here on the handout. So you're basically going to make a table very, very similar to this one, and I want to talk about what's in the table. So these are just very simple descriptive stats. We simply state what they are. So you're not going to actually put error bars on charts or really do anything with the stats. This week you're just going to present the numbers. Later on this semester, we're going to get into what I would think of as kind of real stats, t-tests and ANOVAs and things like that. But for now, just a descriptive data table. So in your data table, you'll have your control, your 2 experimental conditions. You'll list the raw data. What's your raw data? You'll list your N number. N simply refers to the number of replicas you did. So if you did 3, you'll put 3 there. If you did 5, you'll put 5 there. Mean is the same thing as average. And the one thing that might be new to some of you is standard deviation. So if you're new to standard deviation, I wrote out on the handout the formula and a sample data set, and I did the math out for you. And you can see how the data is used by that formula to make a little bit more sense of standard deviation. Probably not going to have a lot of data, so it would be pretty simple to do it at longhand. If you're familiar with some kind of a computer program or some kind of an app that does it for you, go for it. Use that. That's fine too. I just want to make sure you do have standard deviation numbers there. In general, the lower the standard deviation, the better your data is. Okay. All right, so you're going to have that stats table, and I just alluded to the fact that you're going to do a poster. So at the bottom of the last page there is a couple notes about the poster session. So next week, instead of coming to Science Center 121 where you're sitting as you're watching this, you're going to go to Zelnick Pavilion. So Zelnick Pavilion, if you're walking across campus, if you're walking, so I'm used to the science building, so if I'm in the science building, I'm walking over to Houston for something, you'll pass on your right-hand side. It's a glass building. It's by the theater. You'll see a sign. You'll probably see us. It has, again, glass walls, so you'll see us inside with our posters. Just come directly there at 1:20, and we'll have a poster session. I'll have easels set up with poster boards on them, and I'll have some snacks to make it a fun poster session. And you're going to put up your poster, and I'll come around and look at what everyone did. And you'll present your data, present your stats, talk about what worked well, how your Planaria reacted, what you would do different next time. The rationale for doing this is that when people actually present their data, they often do it in the form of a scientific poster. So if you get involved in a research lab at Wesleyan, there's a good chance that at some point you'll get to go to a big conference or a big meeting. But in order to do that, you'll have to take a scientific poster to present. And so again, you'll make a poster about what you've been doing in your research lab, and you'll present it. So I want to give you some experience making a scientific poster and then also presenting a scientific poster. So you're going to make one poster per group. So everyone in the group will work together on the poster. Divvy things up however you want. Maybe someone wants to write up the intro. Maybe 2 people are going to work on making the figures and doing the data analysis. Maybe someone else is going to, I don't know, make it aesthetically pleasing and kind of help format everything at the end. Work together, see what works best for your group. But again, you are responsible outside of class for preparing your poster. A poster is basically like a lab report, but it's a visual lab report. So there's not going to be as much text, but have great visuals of your data. So don't just list the data. You'll want to make, like I have at the top, some kind of a column graph, a bar chart. So visually display that data so that it's easy to see and that it's easy for someone who's looking at your poster quickly to make sense of what's going on. I'd also encourage you to maybe take some pictures that you can put on your poster. Again, visuals just, they always say a picture can say 1,000 words. So make your poster very visually pleasing. One group per poster, everyone should help with the poster. And there's more details about putting together the poster on page 30 of the lab manual. So check out page 30 for even more details. All right. 2 final notes before you head down to the lab and get started today. One is that there's a worksheet, an experimental design worksheet on page 28. Fill that out ahead of time and run it by your TA. Basically, we want to make sure you have a good control. You've thought about how you're going to prepare your solutions before you just start and then realize something's off and you have to start over again. So again, we're not going to collect that worksheet, but jot down some ideas, run it by your TA, and then go for it. Actually carry out your project. The other thing I want to mention is that I want you to have fun, explore a new parameter, but know that it may or may not work. You might think you have this really cool idea. You're going to test, I'm going to call it chemical Z, whatever chemical Z is. And so you have your control, you have a little bit of chemical Z, and then you have a lot of chemical Z. Those are going to be your three things. And maybe the worms could care less about chemical Z. There's no trend. They just swim around at the same rate the whole time, whether or not that chemical is there. And you might be really disappointed because you really wanted to maybe have a nice graph where it starts at 0, and then you add a little bit and it goes up, and then you add a little bit more, it goes up, and you were hoping for these really pretty results and you didn't get it. What you get is kind of a flat line. So control, condition 1, condition 2, they're all the same. That's still data. That's still doing science. So no matter what, you're going to have data, and you'll present your data in the poster. Okay. All right. I think that's all. I hope the lab goes well. Again, if you have any questions, feel free to send me an email. Feel free to stop by later in the week. I'd be more than happy to help you with data analysis, talk you through anything you might need to understand to help you put together your poster. All right. Bye.